presentation. Okay. I'll be giving a, I'll be giving a presentation entitled Directed Induction of Functional Multiciliated Cells from Human Pluripotent Stem Cells. This movie is a side view of murine trachea, which has airway multiciliated cells recorded by high-speed camera. Let me introduce myself shortly. My history almost falls within this area of Japan. I was born and raised in Kyoto City, which is famous for beautiful historical architecture. For example, Kinkakuji Temple and a 5-story pagoda, which is both a national treasure. After getting PhD, I started to belong to Tsukita Labo in Osaka University as a postdoc researcher, and also belonged to the Department of Respiratory Medicine in Kyoto University as a respiratory physician. When I worked for Tsukita Labo, I met Elisa-san and learned a lot while working with her. Today, my presentation is divided into 3 topics. First, I'd like to show you what iPSCs are. Then, I will show my previous research in Goto Labo in Kyoto University about the induction of functional airway multiciliated cells from human iPSCs. Finally, I would like to show some data regarding future application of this method. Induced pluripotent stem cells, iPSCs, are a new type of pluripotent stem cells first generated in 2006 in mouse by Dr. Yamanaka's group. They generated human iPSCs in 2007 also. iPSCs were established by introducing a small number of genes into ordinary somatic differentiated cells. The process of changing a cell from a differentiated to a pluripotent state is called reprogramming. The method developed by the Yamanaka's group has been shown to be highly reproducible, relatively simple, and is considered a major scientific breakthrough. As you know, Dr. Yamanaka got a Nobel Prize in 2012. iPSCs proliferate indefinitely and are thought to be differentiated into any type of cell in the body. It is thought that iPS cells will be useful in elucidating the cause of disease, developing new drugs, and in cell transplantation therapy and other forms of regenerative medicine. Regenerative medicine is a therapy aimed at restoring function lost through disease or injury. This is a schematic for human iPSC-based disease modeling. Human iPSCs are derived from individual patients and differentiated into specific cell types. To develop new therapies, the resultant cells are used to observe disease-specific phenotypes and identify novel pathological mechanics. Human iPSC-based disease modeling with patient-specific cells now provides a new approach for the development of personalized diagnosis and medicine. One example is diseases which arise due to changes in the brain, as brain cells are extremely difficult to acquire and study from live patients. Using iPSCs, researchers hope to compare healthy and diseased cells. iPSCs will also make it possible to evaluate and test pharmaceutical efficacy, side effects, and toxicity in a way not possible in the human body. This is a schematic for human iPSC-based cell therapy. Human iPSC-based cell therapy development usually includes the following steps, as in this scheme. Step 1, collect somatic cells from patients and culture somatic cells from affected patients. Number 2, reprogram patient somatic cells into iPSCs. Number 3, use genome editing technology or viral transduction methods to, to repair patient iPSCs and turn them into genetically corrected iPSCs. Number 4, differentiate the corrected iPSCs into desired cell types to serve as genetically matched healthy donor cells. Number 5, perform quality control tests for cell identity, purity, activity, and safety. And number 6, transplant the genetically matched healthy cells into patients for cell therapy. Regenerative medicine, in the case of diabetic mellitus, for example, involves transplanting cells with the ability to regulate blood sugar level, or in the case of trauma, where nerves have become severe, transplanting nerve cells that can help restore the interrupted connection. Next, I'd like to show you our way of the induction of multiciliated airway cells from human iPSCs. I conducted this research at Goto Labo in Kyoto University. This is Goto Labo. The respiratory system is classified into the proximal airway and distal airway alveolar regions. In today's talk, I would like to focus on the proximal airway in particular. The airway has a tubular structure lined with airway epithelium on the inside, and the airway epithelium has a pseudo-stratified structure and consists of several cell lineages. The multiciliated airway cells, goblet cells, and club cells cover the surface, and the basal cells are present near the basement membrane. It is known that neuroendocrine cells are scattered in the airway and clustered at the branch points of bronchi. Multiple ciliated airway epithelial cells, which I will refer to as MCACs, have a pivotal role for mucociliary clearance in host defense of the respiratory system and are associated with various refractory respiratory diseases, such as primary ciliary dyskinesia and cystic fibrosis. However, it has been challenging to recapitulate human airway diseases in vitro because of the difficulty in isolating primary bronchial epithelial cells from diseased patients before the cells are affected by therapeutic agents and the environment. Generation of induced pluripotent stem cells has opened the door for creating patient or disease-specific pluripotent cells with the potential to differentiate to disease-relevant cell types. To use such iPSCs to model lung disease and to use for regenerative medicine, an effective differentiation protocol to generate airway epithelial cells and functional analysis method is necessary. The manuscripts from other laboratory, when I conduct the research, describe differentiation protocols to generate airway epithelial cells from human iPSCs. However, the induction efficiency of HPSC-derived airway epithelial cells in their reports was low or unclear, and the ciliary movement of HPSC-derived MCACs has not yet been reported. So I conducted this research. Developmental studies in mice and humans shown that lung cells in both the airway and alveolar regions originate from NKX2.1 positive ventralized anterior foragut endam cells. And in our previous study, we identified carboxypeptidase M, CPM, as a surface marker of NKX2.1 positive ventralized anterior foragut endam cells, VAFECs, and demonstrates the potency of CPM positive VFACs to differentiate into alveolar cells. So we hypothesized that airway epithelial cells, including MCACs, could be induced from CPM positive VAFECs from HPSCs, as all lung epithelial lineage cells have been reported to be differentiated from NKX2.1 positive VFECs. I'd like to show you quickly about the earlier stage of differentiation. At first, undifferentiated iPSCs were seeded after single-cell enzymatic dissociation on D0 and were differentiated into SOX17 positive definitive enzyme cells with Wnt agonist and activin A during 6 days of the step 1 period. Next, the definitive enzyme cells were directed to FOXA2 positive, SOX2 positive anterior forward enzyme cells with the inhibition of TGF-beta and BMP signaling during 4 days of step 2. Then, the concentration of Wnt agonist, BMP4, and all trans-retinoic acid was optimized depending on HPSC lines to differentiate the anterior forward enzyme cells into NKX2.1 positive, FOXA2 positive VFECs during 4 days of step 3. Because proximal airways develop as 3D branching structure in vitro and 3D culture of HPSC-derived endodermal cells has been successful in various organ studies, such as the intestine, liver, and alveolar type 2 cells. We adopted 3D differentiation protocol from CPM positive VFECs to induce proximal airway epithelial cells. On day 14, CPM positive VFECs were isolated and matrigel-based 3D culture without feeder cells was started. In step 4, the spheroids grew larger and some of the spheroids began to fuse by day 28. Importantly, confocal immunofluorescent imaging studies shows that nearly all the cells forming spheroids were SOX2 positive, NKX2.1 positive cells, indicating that these cells were of proximal airway epithelial progenitor cells. At the end of step 4, no MCACs were observed, which prompted us to hypothesize that there might be another step for inducing MCACs. So we switched the medium to step 5 medium. After the trial of various conditions of medium, on day 42, cultures of MCACs were observed by hematoxylin eosin staining. Confocal immunofluorescence imaging studies revealed acetylated tubulin positive, oxygen positive cells as observed in the fetal human lung. Confocal immunofluorescence study revealed closely aligned acetylated tubulin positive cells and MAC5AC positive cells as observed in the human airway, while secreted MAC5AC markedly accumulated in the closed lumen of the HPSC-derived spheroids. A small number of SCGB1A1 positive cells, club cells, KRT5 positive cells, basal cells, and PGP9.5 positive and synaptophysin positive neuroendocrine cells were also found. Nearly all of the HPSC-derived PAECs expressed NKX2.1 positive cells. SNTN was localized at the chips of multiple cilia of HPSC-derived MCACs, as was seen in vivo. Next, the morphology of HPSC-derived MCACs were examined using electron microscopy, demonstrating multiple cilia originating from individual basal bodies on the apical surface of columnar epithelial cells. Cross-sectional images of the cilia revealed 9+2 structure, consisting of 9 doublet and a central pair of singlet microtubules, in addition to dining arms, which is a specific feature of motile cilia in MCACs. In light microscopy, beating cilia were easily observed in the lumen of the spheroids and recorded by high-speed camera. And the metachronal wave-like synchronized beating of the cilia was observed in some MCACs. The inhibition of notch signaling pathway induced MCACs And we could generate MCACs at more than 70% induction efficiency in various HPSC lines. This result is consistent with the studies of genetic murine models. In order to quantify the mucociliary flow over the MCACs, we established a protocol of passaging HPSC-derived MCACs in proximal airway epithelial cell spheroids to air-liquid interface condition due to the difficulty in measuring the flow rate inside the 3D spheroids. On day 15-6 of the 3D air-liquid interface protocol, serially beating was observed on the apical side of MCACs, and CFTR was detected in the apical surface of MCACs. The serially beating frequency was calculated after acquiring bright field images of MCACs in the spheroids and the 3D air-liquid interface condition. The serially beating frequency of HPSC-derived MCACs in the 3D air-liquid interface protocol was near the normal serially beat frequency of human MCACs, which range from 10 to 40 Hz. To measure mucociliary transport, the fluorescent beads placed on MCACs was placed in both HPSC and normal primary cultured airway epithelial cells. However, the values appear to be affected by the lack of synchronity of serially beating for generating unidirectional flow. There is no unidirectional flow in the culture. Therefore, we analyze the diffusion of the beads from their trajectories based on the concept of the previous report. We define the diffusion coefficient normalized to Brownian motion as a mucociliary transport index. Then the MTIs in each HPSC and normal primary area epithelial cell derived multiciliate AI cells were calculated, and all the HPSC derived cells showed significantly greater mucociliary transport index compared with Brownian motion. In summary, we established a 3D differentiation protocol for generating proximal airway epithelial progenitor spheroids from CPM positive VFECs. Proximal airway epithelial progenitor spheroids could be induced to generate MCACs and other airway lineage cells without alveolar epithelial cells. The HPSC-derived MCACs have modal shear with a 9+2 microtubule arrangement and lining arms, capable of beating and generating flow for mucociliary transport. These results are published as an article, and currently we applied this method to disease modeling of airway ciliary genetic disease, primary ciliary dyskinesia. We could find disease-specific phenotype of MCACs as ciliary beating pattern in disease-specific gene knockout human iPS cells, derived cells. So we'd like to develop this research to invent new therapy or diagnosis method. Yeah, thank you very much for hearing my presentation. Thank you.
Thank you very much, Konishi-san. So you have demonstrated the efficient and established method for 3D differentiation of to generate PA. There are so many like abbreviation here. PAEC, proximal epithelial cells.
Thank you.
Human pluripotent stem cells. Okay, thank you so much. So last week, actually, I gave a little introduction about differentiation cells and also air-liquid interface, hoping that the students can understand more with this topic. So your protocols are a lot more complex than the generation of MTEX, of course.
But MTEX is also very difficult. So I think,
this one is a lot complex, and you have steps using 2D and 3D. So I think that there will be many questions from the students. Okay, so
Yeah, please ask me.
Oke. Kepada seluruh mahasiswa tadi sudah mendengarkan presentasi dari Dr. Konishi. Kemudian mudah-mudahan ini ya sedikit bisa paham ya, karena tadi saya lihat banyak istilah-istilah memang. Tapi minggu lalu kan sudah ada pengantar tentang diferensiasi sel dan juga tentang air-liquid interface, bagaimana membuat dalam kultur 2 dimensi, gitu sudah pernah kita bahas pada minggu sebelumnya. Jadi apakah ada pertanyaan? Kalau ada pertanyaan silahkan langsung pakai mikrofon atau bisa pakai chat box. Okay, Konishi-san, we're waiting for the questions. Now it's
okay. Sorry.
Are you okay?
Okay, sorry. Yeah.
Is there any questions? Tesla, Pipin.
Halo Bu Elisa.
Who's speaking? Oh, Pipin. Okay, Pipin. So
Yeah.
one student, her name is Pipin Agnesia. I think she would like to ask a question.
Okay. I want to ask something about the explanation. What is the function of BMP4 in the differentiation of NKX2 min1 plus FOX1 plus VCS?
Yeah, she's asking about the role of BMP4. You have several. I asked them to read your paper. So
okay,
I think they found it from the paper about that.
Yeah, thank you very much for asking me. We selected reagents by by checking the previous report of murine development of lung, and in developmental stage of lung, there is BMP4 expression in dorsal side of body, and it will interact with lung differentiation. So we followed the previous report about murine development. So yeah, we know in the developmental stage, NKX2.1 positive ventralized anterior forward tendon cells was differentiated from anterior forward tendon cells by BMP4 concentration change, and we follow that previous report. Could I answer your question?
So basically, well, you refer to the previous report already that the BMP4 is very useful.
In previous report of murine development,
And it's applicable for the human-derived pluripotent stem cell as well.
We didn't know before we tried, but after trying, we could know it is applicable. And we also checked the condition without BMP4, and it cannot differentiate anterior forward tendon cells to ventralized anterior forward tendon cells. So
it's very useful, and also you have to check several concentration. Which one is the optimum concentration?
Okay. That's right.
Oke, Mbak Pipin, terima kasih. Tadi jawabannya seperti itu.
Okay, thank you, Mr. Satoshi and Bu Elisa.
Thank you for asking me, Pipin. Thank you.
Okay.
Is there anyone else? Dian, maybe?
Yeah, Bu Elisa, I like to ask something.
Thank you.
CPM plus cells showed higher gene expression of multisylated cell markers compared to CPM cells. What is the role of the peptides?
Yeah, she's asking about the role of CPM carboxypeptidase, because in your result it shows that the CPM plus plus cells, they can generate or differentiate into more multisylated cells, but CPM negative cannot.
So. As it is a very important question, I think. Thank you very much for asking me. And I sorted the CPM positive cells and negative cells by flow cytometric analysis, and I applied both cells to same culture condition. And in CPM, if we apply the CPM positive cells to our culture condition, we can differentiate multi-ciliated area cells. But when we apply the CPM negative cells, we cannot differentiate to multi-ciliated area cells. I think this is because now, to be honest, the work of CPM is not to be elucidated well, but we use the CPM as a surface marker of NKX2.1 positive ventral anterior faggot endowed cells, which is progenitor cells of airway and alveolar epithelial cells. So by sorting CPM positive cells, we can sort lung progenitor cells and we can differentiate cells. So if we selected the CPM negative cells, it is not lung progenitor. It was not lung progenitor cells, so we cannot differentiate such cells into lung multi-ciliated area cells. I think it is a very good, important question. Thank you for asking me. Yeah.
Okay, so Diane, so that's the answer. So basically, simply it's because the CPM plus is the marker for lung progenitor cells, right? So that's why it gave rise to the whatever epithelial cells derived from lung progenitor, multiciliated cells, neuroendocrine cells, and club cells and other cells.
Yeah,
yes.
Yeah, that's right. That's right. Thank you.
Thank you.
Okay, thank you, Mr. Konishi and Mrs. Elisa.
Yes, thank you. Oh,
thank you.
The question from the chat box is from Ibu Ari Susilowati. She's a lecturer. She's a staff, faculty staff. So her question is the ciliary cell can defect by genetic or any other factor? So you need to improve or repair the ciliary cell.
I checked messenger RNA of differentiated ciliated cells, and also we checked single cell messenger RNA analysis, and there is some difference between real lung cells from human adult and iPSC derived air epithelial cells, but the expression level was nearly same. So I think regarding multi-ciliated area cells, we could differentiate nearly mature cells in human adults, and we also checked functional analysis, and it was nearly same in human adults. So regarding when we think about the single cell level, it might be good differentiation protocol, but we need to make multi-ciliated area cell sheet to model disease, and there is intracellular coordination of ciliated cells in real trachea and airway, but we could not achieve that because in cell sheet the ciliated cell was not interciliary coordination was not found, so we have to improve that system. But regarding single cell level, messenger RNA expression and protein expression and function analysis was achieved, we think. Could I answer your question? I'm sorry if I couldn't understand.
Konishi-san, I think what Dr. Ari asking is, I'm sorry to interrupt, is a more general question, I think. She is asking not about your protocol, about the ciliated cells in our body. If we found a defect on the ciliary cell in our body, was it caused by genetics or caused by other factors, for example, lifestyles or something? And if you find, like you explained about primary ciliary dyskinesia, is that a genetic matters or affected by infection or something? And if it happens, like if there is a ciliary defect, how do you, what to do with that? I mean, you repair it or you replace, I don't know, you're a doctor, respiratory physician, so that's
thank you very much for asking me very important question. And there is a genetic disease of ciliary multiple ciliary airway cells. As Elisa-san said, primary ciliary dyskinesia is a genetic disease of cilia. Cilia has 9+2 structure of microtubule in each cilia, and primary ciliary dyskinesia show about 40 different genetic defects, and each genetic defect will affect the ciliary structure in each cilia, and it will affect ciliary movement, and it will affect the mucociliary transport, which leads to disease. And also, as you pointed out, for example, COPD or asthma, which is a very common disease, can affect ciliary structure by after the birth, and so it will also reduce ciliary movement in final. So in real practice or medical field, there is no replacement treatment about ciliary defect, but so we can only promote the mucociliary transport by changing character of mucus. We can only do such a thing, but in the future, if we can conduct cell therapy, cell treatment therapy, we can replace cells and proliferate cells and transplant cells, or we can add, for example, virus vector to some patients which has ciliary defect and make rescue the cells of stem cells, basal cells, stem cells of multiciliary airway cells, and make good multiciliary airway cells, but it will take much cost, so we have to balance between cost and efficacy, and it is a very difficult thing. Yeah, it is a very important question. Thank you very much. Yeah.
Thank you very much. So
okay, thank you.
Yeah, thank you, Ali.
It's very clear in the second explanation.
Thank you very much. Thank you very much.
So at the current technology, maybe the doctor will prescribe just medicine to like reduce the thickness of the mucus, right? To make the patient like easily remove the mucus. I don't know. That's what the doctor prescribed to me because I also have some allergies, so respiratory allergy. Okay, we have also question. Good morning, Dr. Konishi. I'm Rizka.
Okay.
I'm Rizka Meisari, and I have question. When applying to the patient step, is there any serious side effect could happen, and what is your biggest obstacle while do the experiment until the final step?
It is.
What she asked is in the contents of iPSC.
Okay.
Yeah, okay. If you can answer.
Yeah, thank you very much. It is a very also important question. Yeah, most biggest problem we have to challenge is occurrence of tumor. iPSCs can differentiate each organ specific cells, but not perfect, not 100%. And there is some undifferentiated cells. It will be differentiated to teratoma, and it will make tumor. So many researchers is conducting research how to reduce tumor making, and there is some obstacle also. So we have to elevate the efficiency of differentiation and how to remove tumor. But in some clinical trials, they could succeed in removing tumor in various ways. And in my side, what most biggest obstacle is how to, there is two applications. One is disease modeling and one is cell treatment. And disease modeling might be easier than cell treatment because we don't need to think about tumor making or the cell activity in our body. So when we think about disease modeling, it is easier. But when we think about cell treatment, we have to think about tumor occurrence and also how to engraft cells to our body. It is also very difficult obstacle, and we could not clear that obstacle. It is our challenge in the future. Yeah, thank you very much. Yeah.
So in the field of medical, in the medical advance, so currently the tumor thing is still a big problem. Detection of tumor.
Some researchers could succeed in remove tumor, but in various ways. But we have to always think about that.
So Konishi-san, I'm sorry, there are still many questions. You still have time.
Oh, okay. Of course.
Okay. So from Aurora Diaz Anasia.
Okay.
Thanks for your time, Mr. Satoshi. I want to ask, will there be any development of testing to human, or is it focused on test animals, and what should be considered in cilia cell culture? I think this question is related to your previous explanation about the future application in human.
I see.
This
Yeah.
technology.
Currently, we have to test animals when we develop drug or clinical trials. But in some research, there is a big difference between animal model and human. So in some researchers conduct skip the animal trials using iPSCs. For example, in brain field, in some Japanese researcher conducted clinical trials without animal model. And if we establish good system using iPSCs, we can skip animal trial in some case. But it may be it may take more time. Could I answer your question?
Bagaimana Mbak Aurora Diaz? Sudah terjawab belum? Okay, well, if there is something more, so I think she can respond later, maybe.
Okay, thank you.
So let's go to the next question.
Okay.
This is interesting from Mumtaz Azaki. Thank you, Mr. Konishi, for your explanation. I am Mumtaz from Bioscience Department. I would like to ask the question, is there any particular epigenetic factors that prove to be challenging factors in developing human induced pluripotent stem cells? So he's asking about whether is there any epigenetic factors influencing this HIV.
In my way or in general? Okay.
Yeah, I think both you in your experience, or you may encounter from other researchers' experience.
To be honest, in my way, I haven't checked the epigenetic factor yet, but it is future experiment we have to do. And in other organ field, some researchers check the epigenetic factor, as you said. And in some research field, I'm sorry, I couldn't say in detail, but some epigenetic factor will affect the differentiation efficiency or maturation of differentiated cells. So as you think, it could be important factor, but in my case, I haven't checked. It is a future task for us. Thank you.
Okay, thank you, Konishi-san. So you haven't checked the epigenetic, whether epigenetic factor influence or not, but maybe epigenetic is very wide. I mean, it can be anything. So it could be influencing.
I think so.
And this is from Risma Hernanda. I think this is related to your protocols in your paper, because you added the DAPT. From day 28 until day 42. So is it critical to the addition of DAPT during that period?
Thank you for reading my article before. Thank you very much. And it is very good question. And notch inhibition will affect the developmental character in Murin Lang previous report. And DAPT will affect the efficiency of differentiation rate of multiple CI exterior cells. And if we did not add duct, we can differentiate multiple airway related cells also. But if we add duct, we can elevate the differentiation efficiency regarding multi-related airway cells and neuroendocrine cells. And it is consistent with the previous report of murine developmental study. So it is not critical. Without duct, we can differentiate, but we can elevate efficiency. So when apply the current study, we are applying to disease model, and we are adding duct every time because we'd like to elevate percentage of multiple ciliary airway cells. But if we think about the real body, there is defined composition between cell lineage, and if we add duct, the result will be changed from real body. So if we think about the cell interaction between other cell lineage, it might be better not to add duct. But when we think about now current project, we'd like to see high efficient multiple ciliary airway cells, so we are adding duct. Yeah, but in the future application, it might be better to exclude duct. Yeah, it is a very good question. Yeah.
So the duct, I mean, without, you said that without duct addition, so it actually can generate multiciliated cells by itself, right? But not not enough number, not enough proportion number.
Proportion. Could you say that again?
Yeah, the number of multiciliated cells is not enough, so that you need to add duct.
Yeah, if we'd like to analyze mucociliary transport in disease model, we'd like to see many multiple ciliary airway cells. So we elevate, but in real body, the composition will be different, lower percentage. Yeah, and as you asked me, the earlier timing of adding duct was also checked by us because in previous report, notch emission in early developmental stage will change the rate of proximal airway and distal airway. And by adding duct, we can elevate distal airway and alveolar region relative cells. So we tried, but we cannot get good results. So we only add later period in this project. Yeah, it's a good question. Thank you.
Yeah. Thank you. So this is probably the last question from me.
Okay.
I'm curious about the way you create. You show from one slide there is a ALI in 3D.
Yeah.
But to my understanding, ALI is the air interface. So how do you create ALI from spheroids?
ALI. I'm sorry, my explanation was, but I'd like to show the difference of method between ALI and 3D culture. And ALI is the same method as you conducted in MTEX. And 3D culture, we also use transfer. And we added 50% matrix gel with 50% medium, including cells, on transfer. And we make gel on transfer, and we added medium surrounding transfer. Yeah, as we conducted in ALI. Yeah.
So the cells are still submerged in the medium.
Cells are submerged in gel.
In the gel.
In the gel, and from lateral side and bottom side, medium can interfere the gel.
I see.
Yeah.
I see. So that part I missed. Okay.
No, thank you.
Thank you very much, Konishi-san. I think that's all for the questions, and maybe wait.
Okay.
Oh yeah, just thank you from Rismaira. Thank you for the answer, sir. It's good explanation. She understood.
Thank you very much. Yeah, so, thank you.
Once again, thank you very much, Konishi-san. So it's very great. It's been a pleasure for you to join us and share all your experiments here. I I hope next time we can join in again in other opportunity, maybe.
Yeah. I love it. Yeah, thank you very much for giving me very good opportunity to present my work here. And thank you for asking me many questions. I was very pleased because you looked into my research. Thank you very much. Yeah.
So I would like to close this session in Indonesia. So,
Okay.
wait. Terima kasih semuanya. Jadi ini sudah akhir dari kelas ini ya. Memang tidak bisa lama-lama karena di sana juga sudah malam. Jadi terima kasih sekali sudah bergabung dalam kelas Kultur Sel dan Jaringan Hewan untuk topik spesial pada pagi hari ini. Ini ada pertanyaan dokumentasi enggak? Oh enggak, tidak ada dokumentasi Mbak Maria, Mbak Dian karena sudah saya record ya. Ya. Kemudian jangan lupa untuk presensi untuk mahasiswa. Ya, sekali lagi mudah-mudahan apa yang kita dapatkan hari ini sudah bisa bisa bermanfaat bagi kita semua. Gitu, Mas. Oh, I'm sorry, it's a long closing.
No.
Okay, thank you, Konishi-san. Have a good day and good luck with your research.
Yeah, thank you, Elisa-san. Yeah, have a good day and good luck.
Thank you.
Thank you. Thank you.
Satoshi and Bu.
Ya, terima kasih. Thank you.
Thank you.
Have a good day.
Have a good day. See you.